Hi everyone. In this video, I'm going to talk to you about how you can determine for genome-wide association study which build you have. So, for instance, let's look at this genome-wide association study here for bipolar disorder. Oh, do you want to go? You can go, little guy. Bye, little guy. He's very cute. So there's a genome-wide association study that has identified, was just published in 2021, and they have sort of identified more than 40,000 bipolar cases of bipolar disorder. And bipolar disorder has a lot of side effects and a lot of things that people go through and experience. So just to show you guys, about bipolar disorder. So it's, used to be called manic depression. I think my computer is a bit maxed out. Okay, so it's a manic depression. It's a mental health condition that causes extreme mood swings that are, emotional highs, mania or hypermania, when you're emotionally high in a very happy state, and lows that like depression. So when you become depressed, you may feel sad or hopeless and lose interest or pleasure in most activities. So this is an overview on bipolar disorder here. There's some overlap with borderline personality, but some key differences between borderline personality and bipolar disorder as well. So, you have like depressive, which is negative emotions, feelings of worthlessness, hopelessness, guilt. Low is the you don't have low energy, low appetite, low interest and pleasure, and physical sufferings, constipation, fatigue, and insomnia. And so that's your very low stage. And then your really like manic stage, happy stages, your mood can be irritable, expansive, elevated. Your speech can be loud, rapid, running, and poor judgment. And physical sufferings can be you're distracted and hyperactive. So there are a lot of symptoms that are also associated with it too. So it's like two different types. So the manic episodes, feeling overly happy for long periods of time, talking very fast with racing thoughts, becoming easily distracted, having overconfidence in abilities, and engaging in risky behavior like gambling. So these are the manic stages. And then there are also depression episodes. So there are lots of highs and lows. So it's like a roller coaster. So you can feel sad or hopeless for long periods of time, have a significant change in appetite, think about or attempt suicide quite a bit, which is really unfortunate, feel fatigue or lack of energy, and have problems with memory or concentration. So that's just an overview on bipolar disorder. So here is this genome-wide association study that was published on 57 bipolar disorder cohorts in Europe, North America, Australia, and they have 41,917 individuals with bipolar disorder, and these many controls. They also have type 1 and type 2 that they're measuring as well. This is a major histocompatibility complex. A lot of our immune system genes come from this region on chromosome 6. This is the MHC region. So we can see again, negative log 10 p-value in chromosome. So when you actually load this in, so let me show you, when you load this in, what it actually looks like is so when you load this in with this file, so here I'm just sort of reading in this file here. This is I'm focusing here in this example on bipolar 2021 genome-wide association study type 2, which came from the same paper, and they mentioned that they had 6,781 cases and these many controls. And when I read this in, what it looks like is it has, let me just, portray it again. As a chromosome number, position, and ID, this is a SNP ID, single nucleotide polymorphism ID. So essentially, this information here, on the, let me also just show this again. Okay, so it'll be custom. So this chromosome and this position information can be used to then just map to this genome-wide association study Manhattan plot, which looks like the skyscrapers in Manhattan. So you can go from this chromosome number, which along here, and these are positions along the chromosome, scanning along the first chromosome is in blue, then red is the second chromosome, blue is the third chromosome, red is the fourth chromosome. So alternating colors to help you distinguish between each chromosome, but this is sort of like going along the base pairs in a chromosome. So what happens is, how do we determine this reference build? What reference build is this information? So we have all these SNPs here and this information. This is just the head. If you see the SNPs file is about 7,188,236 rows. So DIM of SNPs file has so many rows in it and a lot of columns. So how do we know the build? Sometimes they don't really mention it. So I could look up here and you have a hunch. If I can look up Hg18 and then I can see, okay, it has Hg19, it doesn't have Hg38. It doesn't have that. So it seems like it could be Hg19. So I'm going to show you how to do that, then I can explain the rationale afterwards. So what I'm going to do is I'm just going to look at this information here. Let me just actually go to Caviar. So this is, I have videos again about looking up in Caviar. So you can put in SNP IDs, you can put in a chromosome number and followed by position. So you can put in anything. So basically, what I've done is I've looked at this head of the SNPs file, that's just the first few rows. And I just focused on taking just a few rows so that we can figure out what is the reference build, because that could be very important, especially when you're mapping coordinates. So I sort of just took the first few ones, like this one corresponds to this one here. And then the next thing you'll see is that, let me actually put it more like this here. Is the top one. Then I also wanted to get a little bit of both. So I also got the chromosome number, which is X chrome, and then the position here. And then similarly, you'll see that I also did the same thing. Like I just got a few of the ones from here. I just kind of took this information, like for instance, RS 11727821. That's this one here. Then I also got the corresponding position, chromosome 8, position 1005, 16008. That's what this is over here. And then I just, for the remaining, I kind of just didn't want to make it too long. And just give you guys an idea. You can be as extensive and as thorough as you like, but you can just have this like this here, and you'll have like, you can put the chromosome numbers, you can put the RS, the SNP IDs. So what we're going to do is you can put as many of these as you like. You don't just have to stop at just these. You can put all of these, the millions, and not millions, it's actually capped at 100,000 that you can do in each run, but you can just put a few in because you'll see my point. So if you don't know the reference build, what you can actually do is you can just take a few examples. You can put chromosome number and positions like I did here. And in this example as well, you can take those numbers, you can put the RSIDs there, you can put both. And we're just going to see which of the reference builds has this information. So there are these two reference builds that Caviar allows you to look at, Hg19, GRCh37, and then there's Hg38, GRCh38. So firstly, let's just look at Hg38, which is the more modern ones. So we just have a few examples and we're just going to look at that. And let's see how the results kind of fare. So, okay, RS11278216. This one. Okay, so the first output is giving us is for this, and this maps with this here. Okay, and the corresponding position in Hg38 that it's found. So again, this is Hg38, which is also known as GRCh38. What it's found is the following. It's instead said that this is chromosome 8. Okay, yep. But what a position along that it found, I mean, the chromosome number should typically be the same. It's just the position along it that may differ. 9950378. That does not match this. So this here, it does not match. That's a concern. So this here and this here do not even match. Between these two here, it's not a match. Uh-oh. So that one also, so you can kind of see, maybe it's not Hg38 because, this one didn't even match. Then the next thing that we did is if we tried looking this up, chromosome 8 position, the same position, we tried looking this up, and what it told us is it's not found in Hg38, but it's found in Hg19. So we had taken those positions and it's not even found there, and it told us that, hey, it's not found here, but it's found here. So that kind of tells us that the reference genome that was used for this data set is probably Hg19. And to just make it, very thorough, just to also convince yourselves a bit more. So this RS6251385, this one here, it's supposed to correspond to a position of 1015. Let me just remove that. It's supposed to correspond to a position of this, the SNP here. Just to show you, let me just put a star here. It's supposed to correspond, this SNP here, it's supposed to correspond to a position of this, but instead of 10159221, but instead it corresponds to 100579985. So that's also another sign. And in fact, if we try looking up this information, it's going to tell us it's not found in Hg38, but it's found in Hg19. So again, this is not found here. And we can also see the same results as we, scroll along. We can also note that this RS125466060, which should correspond, this one here, this one should technically correspond to, it should correspond here to this position 105178584, but instead it corresponds to this different position. So it just doesn't map at all. And there are just some additional examples here of how, this position here, this top position is not even found in Hg38, but it's found in Hg19. So this is just some additional examples, again, of how they just don't map up. If you look at this one, you see that this position here is not the same as this one for the same SNP. It's just not the same. 107669447 is not the same as 108681675. It's just not the same. So I can also just prove again that it actually works instead, not for Hg38, but we can try Hg19. And now let's just make sure again. So it should work for this. That's what it told us. And if that's the case, then the build is Hg19. So here, now let's check this one out. So we see that this ID here, this is the same ID, RS1172782116. So these are just the SNP IDs. And again, guys, the SNP IDs are just like the IDs of, each of these is given, and a SNP ID is given by an RS number. Each of these has an RS number, which is a chromosome, a number, and a position. And this just means going along chromosome 1, going along chromosome 2 from the start to the end. And these are the corresponding negative log 10 p-values. And we also have p-values in our data set. This p-val column is basically what this is, p-value. And you take negative log 10 of that for each chromosome and position, and you have this Manhattan plot. And there are lots of functions in R that can help you make this beautiful Manhattan plot. It takes a while to make. Okay, so going back to this, we are in Hg19. This is Hg19, which is also known as GRCh37. Oh, 37. Okay, 37. And that's what this is here. And what we can do is just see that, it makes sense what we get. So we were looking at this SNP here, RS1172, this one and this one, they match. Okay, so now let's just look at the position and see that this 8 and this 1005160008 is exactly found here. That's great. So the same SNP ID and the position are found here. And usually the fact that it was found in Hg19 and not in Hg38, the other one, that can be reason for us to know that it's Hg19, but you might just for yourself want to do a few more checks to make sure. So we can check the next one. So we found indeed that, this position here is the same position here for the same SNP. That was exactly the same. So that is great. Okay, 1 second. Let me write this here. Great. So that maps. Now let's just look at the next example here where we see that RS625, so we focus on this one here, 625-13865, and the corresponding position was 101592213, which is exactly what this is here. So those two also match. This maps with that. So that's great. So that's also another sign as well. So that's just how you can tell, okay, it's HG. That's what it is. And then similarly, you can also see again, you can keep doing as many checks as you want, like this SNP here, the corresponding position of this, the same SNP, this one and this one are identical. That's great. We're happy. Yay. Okay, that's us happy. Okay, so that is a happy smiley. So this and this map, and for the same SNP here, this is the same as this. And then they also had this information. I think you can disregard the warning. We also, okay, so this one is 10697 3048, the first one. Great. And it maps to the same SNP, right? Like, so that's great. This same position maps to the same SNP. So that's also just validating, that this all is making sense. So this just goes to show that this was indeed Hg19 that was used. And then this one, again, also works RS9, this one and this one are the same. And then we see that this position 108681675 is the same position here. They're both the same. So that's just great. Awesome. For the same SNP. And lastly, in our example, we can see this. Last but not least, this one here has 108690829, which is exactly what this is. So this maps to this for the same SNP. So again, there's just this connection here between these two and between these two. So that's just great. So that's awesome. And we're loving that. So what we can tell from this information is indeed that, okay. We can indeed tell that this is Hg19. From the SNPs file, okay. I can say source, I can say Hg build is just equal to Hg19, which is GRCh37. I can say that was the original Hg build. The Hg stands for Human Genome. So indeed, now we just added a column that says that that's what the original build is. Okay, so that's all great. But what does it mean exactly? You might be wondering. So I'll show you when that finishes loading. So people are sometimes asked this question, like, how can I identify the genome build of my GWAS data, right? Like, how do we know what it is? And then you might be wondering, that what are the different builds exactly? Successive versions of the human genome reference, commonly called assemblies or builds, have been published since the original draft genome project publication, bringing gradual improvements in quality made possible by technological advances, as well as improvements in the representativeness of the reference genome sequence with regard to historically underrepresented populations. Oh yeah, so after we fixed this, now we added it in, and now our genome-wide association study says the chromosome information and the original build now, which is Hg19, GRCh37. And you would kind of wonder, okay, well, why can't they always do that? Well, the issue is that, like, these SNP files are so huge, so if they added in the column information, potentially it would just take up more room. So we know that this is the build of the mappings. So there are multiple builds. So the latest build of the human reference genome, which is GRCh38, which stands for Genome Research Consortium Human Build 38, which also could be nicknamed as Human Genome Build 38. It can really expand the repertoire. So this is the same over here. This is a carrier type, this is for XX is for a girl, XY is for a male. And these are the chromosomes, from one, these are the main chromosomes that we have. Everyone gets at least one of these from one from mom. So what you definitely get from mom. And in the case of dad, you could get, if you're a boy, you get this. So you get this from mom, and you also get this from dad, except if you get this from dad. Dad gives X for girl and Y for the son. Let me make the color again. And the mom always gives X. So this is just each from each parent, and you can sort of see. So this is all from, like, mom typically just gives, the mother typically gives, mom always gives the X, Mom gives that, and then dad gives everything, but the difference is that dad also gives an X in the case of a girl and a Y in the case of a son. So there are many different assembled, chromosomes, human builds as well that we have, and it's all just the mappings on the coordinate system. So this is sort of what, a reference genome, also known as a reference assembly, is a digital nucleic acid sequence database assembled by scientists as a representative example of the set of genes in one idealized individual organism of a species. As they are assembled from the sequencing of DNA from a number of individual donors, reference genomes do not accurately represent the set of genes of any single organism. So it's like a mosaic. So we're just taking some individuals, of the human, some individuals of some cows, some cats, some dogs, there's so many different types. We're just taking those as just some reference examples of what do we typically expect, like what separates us humans from dogs, from cats, from anything, right? We're just wanting some reference humans. So they don't represent the set of genes of any individual, but they're just giving us some idea. So there's like a haploid mosaic of different DNA sequences from each donor. So we just sort of combine like those, try to find commonalities across some of the donors and try to figure out what is typically like found in humans. And then we have a lot of mutations, a lot of SNPs, single nucleotide polymorphisms, most common form of genetic variation between people that we can sort of then use to determine, well, how do these single DNA bases, because we're so identical, most of our DNA is the same. And a lot of it is really identical with chimpanzees. So we can sort of figure out, let me actually show. So just to show you guys, human identical with chimpanzee. So humans and chimps share a surprising 98.8% of their DNA. So if we share so much DNA in common with another chimpanzee, then we share like a lot with each other as well. You know, humans and chimps, right? You know, we evolved from that, right? So there's so much in common with the chimpanzee that we have. Oh, this is adorable. This is so cute. So we have so much in common with them that it's just those SNPs, those single nucleotide polymorphisms that can help us like determine what causes, you know, ethnicity, like us to look a certain way, have certain phenotypes, hair color, eye color, diseases, whatnot. You know, we can try to analyze regions along the genome, like for bipolar disorder. So what we can see as the cost of DNA sequencing falls. So the human reference genome, GRCh38, is derived from 13 anonymous volunteers. So they kept the identity secret, but they gave it to us, and we could sort of, you know, understand what that is. You know, like what is typical human genome, and this is the most modern one that we have as of 8 years ago. So this is what they're showing. This is the whole, you know, first printer of the human reference genome was printed as a series of books displayed at the Wellcome Center collection in London. So all this information, so many books and books and books of genetic information. So, you know, new full genome sequencing technologies emerge, more genome sequences continue to be generated, and references genomes are typically used as a guide on which new genomes are built, enabling them to be assembled much more quickly and cheaply than the initial Human Genome Project. You know, with the entire genome, James Watson, in regions with high allelic diversity, such as the major histocompatibility complex. This is the immune system, MHC region, a large locus that code for cell surface proteins essential for the adaptive immune system. So like the B cells, the T cells, what is a really new immune system that sort of reacts and is very antigen specific, you know, in cases of attack. So Watson, so many differences while about could not be matched. So anyways, you know, measures of length, as simple as just to count the number of base pairs in the assembly at the genome length. So the mammalian genomes as well, you know, there's so many different human and mouse reference genomes are maintained and improved by the Genome Reference Consortium, a group of fewer than 20 scientists from a large number of genomic research institutes like the European Bioinformatics, EBI Institute, National Center for Biotechnology Information, Sanger Institute, and McDonald Genome Institute at Washington University in St. Louis, fixing mixed representations in the sequence. So the human reference, this is the current one, GRCh38, was released from the Genome Reference Consortium on 17th December 2013, had these many gaps, whereas the first version had so many more gaps. So this one saw the closure or reduction of more than 100 gaps, nanopore sequencing as well. So basically all this showing here is that it's a reference because it's like an indicator for genetic features. And the 1000 Genomes Project is creating a database to provide information about the variations in genomes across the human population. So we have this new one, which is called Hg38. We also have the release called GRCh37, which was released in Feb 2009, which is also called Hg18. So it's sort of confusing, but that's just the name. NCBI had another previous build a few years earlier in March 2006, which is called Hg18. Then this is Hg17, NCBI build 34, and then Hg16, which is NCBI build 34, and this is NCBI build 35. You know, less than a year later, they had like a different build. And then a couple of years later, they had this new one. So what I'll also be talking in another video about how you can kind of go from, you can kind of map between the two. Like if you have one reference genome, and a lot of, in the Genome-Wide Association study we were looking at, it was an Hg19. This was the one that it ended up being in for bipolar disorder. So it ended up being in this region, but it ended up being this one. So we might want to convert, so we can go from one to the other using Liftover analysis. Well, it's called Liftover commonly. And I have a video going through how we can do that in R. So this is Liftover. And you can also go the other way too. You can go down as well. You can go, you can also go in this direction, from Hg38 to Hg19. And that process is just called Liftover. And like, for instance, you could see, sequences of two assemblies like Hg19, Hg38, and, you have a leftover chain file that we'll talk about, and you can perform leftover. So you have a coordinate points or segments, and then you perform leftover to kind of go from the old coordinate system to the new system. So you can go from an old to a new coordinate system here. Like where are you and chromosome Hg19 to Hg38? Like if you're looking at a set of positions, so this is Hg19, the older reference genome here. This is the newer one. And how do these coordinates map to each other? Then this one maps here, this one maps here. Like how do you map these old positions that we now know are Hg19 build? Like how do we go and map this for the chromosome and this RSID? Like how do you map this position from Hg19 to Hg38? So that's what Liftover allows you to do, to say, okay, this match, this is matching. Okay, this one needs to go here. This is a new coordinate system. So that's what it allows you to see. So that's Liftover. It's like figuring out going from a source genome, which is if you're going from Hg19, if you want to convert to Hg38, we have a source genome, we have a target genome, and we're figuring out what are the same, what is different. And all this stuff has been organized for us. And similarly, we also have like mouse reference genome too, so the mouse is called MM. So we have MM6, which was in March 2005, MM7, MM8. And now the most recent one was in December 2011, which is called MM10. So that's just how you can figure out the reference genome, which is really important because genomic coordinates can have very, very different interpretations based on the coordinate system that you're using. So just because a file like the bipolar 2021 genome-wide association study was released this year, sometimes they may not always be in the most latest format, which is Hg38. They could be in the older format, which is Hg19/GRCh37. And then you can do leftover to do the mapping from the old coordinate system to the new coordinate system. So you shouldn't just assume like I had made that mistake myself, and you make an assumption, and that's not always great. So you have to just check, do some spot checks, and you can use the tool I was showing you by Caviar to basically figure out what is the potential, and you can also just infer that, hey, it seems like, even you might have your doubts, but the important thing is that for the ones that we checked, for the SNPs that we checked and the positions, they weren't even found in Hg38. It kept telling us, to check Hg19. So sometimes that's a really good way to know that, hey, for these ones, it's not even found in Hg38. So, and it's found in Hg19. So that could be really good enough, just a few examples to see that the build is Hg19. So yeah, so please don't make the assumption just because it's released this, the current year, that it's going to be the new system. I think a lot of genome-wide association studies are actually Hg19 from what I've been observing, but please don't take my word on it. Please just verify it and just do a few checks, because it's so much better than you going downstream and, making some mistakes. So I'm trying to help you avoid making some mistakes that I had also made initially. So hopefully this helps you. So please let me know if you have any questions, and subscribe if you found this helpful. Thank you.